From the moment we take our first steps to lifting heavy weights, our muscles are in a constant dance of contraction and relaxation. This intricate process is the very foundation of our movement. It allows us to interact with the world, accomplish incredible feats of strength, and experience the joy of physical activity. But have you ever stopped to wonder how this invisible machinery works? How do our muscles, those bundles of fibers, actually contract and generate force? The answer lies in a fascinating microscopic world within our muscle cells. Here we find a highly organized structure of protein filaments that interact in a precise and coordinated manner. Imagine a group of people pulling on a rope. That's similar to how muscles contract, except we are dealing with incredibly small protein strands. This is known as the sliding filament model of muscle contraction. It all starts with a signal from our brains, a command sent through our nervous system to the muscles we want to move. This signal, an electrical impulse, travels at lightning speed along nerve fibers until it reaches a specialized junction called the neuromuscular junction. Here, the nervous system meets the muscular system and the real magic begins. Just as a spark can ignite a fire, this nerve impulse sets off a chain of events that ultimately leads to muscle contraction. At the neuromuscular junction, the nerve impulse doesn't directly stimulate the muscle fiber. Instead, it relies on a chemical messenger, a neurotransmitter called acetylcholine. Think of acetylcholine as a tiny boat carrying a message across a river. The river, in this case, is the synaptic cleft, a small gap between the nerve ending and the muscle fiber. When the nerve impulse reaches the end of the nerve fiber, it triggers the release of acetylcholine into the synaptic cleft. Acetylcholine then diffuses across this gap and binds to specific receptors on the surface of the muscle fiber. This binding is like inserting a key into a lock. It unlocks the muscle fiber's potential for contraction. The binding of acetylcholine to its receptors causes a change in the electrical charge across the muscle fiber membrane. This electrical disturbance is called depolarization. It's like flipping a switch, and it sets off a cascade of events inside the muscle fiber. Depolarization travels deep into the muscle fiber via a network of tunnels called T-tubules. These T-tubules are like internal messengers, ensuring that the signal to contract reaches every corner of the muscle fiber. One of the crucial structures that the T-tubules reach is the sarcoplasmic reticulum, a storage site for calcium ions within the muscle fiber. Calcium ions play a vital role in muscle contraction. They are like the fuel that powers the engine. When the depolarization signal reaches the sarcoplasmic reticulum, it causes the release of calcium ions into the surrounding cytoplasm of the muscle fiber. Now, let's zoom in even further to the level of those protein filaments we talked about earlier. There are two main types: actin, the thin filament, and myosin, the thick filament. These filaments are arranged in a repeating pattern, forming what we call sarcomeres, the basic contractile units of a muscle fiber. Normally, the myosin heads, which are like tiny arms extending from the thick filaments, can't bind to the actin filaments because their binding sites are blocked by a protein called tropomyosin. This is where calcium comes in. When calcium is released from the sarcoplasmic reticulum, it binds to another protein called troponin. Troponin acts like a calcium sensor, and when it binds to calcium, it undergoes a change in shape. This shape change moves tropomyosin away, exposing the myosin binding sites on the actin filaments. With the binding sites now open, the myosin heads are free to attach to the actin filaments, forming what we call cross bridges. Once attached, the myosin heads pivot, pulling the actin filaments towards the center of the sarcomere. This is known as the power stroke, and it's the heart of muscle contraction. Imagine pulling a rope hand over hand. That's essentially what the myosin heads are doing, pulling the actin filaments closer together. The myosin heads then detach, powered by ATP, the energy currency of the cell, and the cycle repeats. This continuous cycle of attachment, pulling, detachment, and reattachment causes the sarcomeres to shorten. And as countless sarcomeres shorten simultaneously, the entire muscle fiber contracts. Muscle relaxation occurs when the nerve impulse stops. Calcium is pumped back into the sarcoplasmic reticulum. Troponin returns to its original shape. Tropomyosin blocks the myosin binding sites on actin, and the filaments slide back to their relaxed position. Understanding the intricate process of muscle contraction is not just a matter of scientific curiosity. It has profound implications for fields like medicine, sports science, and physical therapy. By understanding how muscles work at the molecular level, we can develop better treatments for muscle diseases, optimize athletic performance, and help people recover from injuries more effectively. So, the next time you move a muscle, take a moment to appreciate the elegant dance of proteins happening within your body, a testament to the wonders of biology and the beauty of life's intricate mechanisms. Thanks for watching. Don't forget to subscribe for more videos like this.